Good morning. Today I'm going to explain to you the value of gut microbiome, its disturbance with many kidney disorders. I'm going to discuss this topic under the following sectors: introduction and definitions, what is the correlation of microbiome with different health arenas, gut-kidney axis and its relation to chronic kidney disease progression. Is there a role for gut microbiome to release uremic toxins? The most important point that I'm going to discuss with you is how to manipulate gut microbiome by many interventions, and I'll end with conclusion. I'll start with Hippocrates' statement, who said that death sits in the bowels. This means that the gut is very important. Healthy gut and intestine is crucial for the well-being of a person. If we look at this normal section of the colon from a mouse, normal mouse, by in situ hybridization and fluorescence technique, blue color refers to the normal epithelia, and this is the mucus in green color, and all these are gut microbiome, microbiota bacteria surrounding food debris and fibers. I'll start with some important glossary. What's meant by holobiont? This is a totality of organism in a given ecosystem. This means that as a human being, we are living in a community with bacteria. So the total person with his gut and other non-gut microbiome can be considered as superorganism. Metabolome is the complete set of small molecule chemicals found in a biological sample. Metagenome, all the genetic material present in an environmental sample consisting of the genomes of many individual organisms. If we look at ourselves, we have in the gut bacteria, the genomic material of bacteria exceed 100 our human genome. This means that, and both the human and gut microbiome genome are in a state of evolving together. Microbiome is the collection of genome of microbial environment or ecosystem. Microbiota, the microbes that collectively inhabit a given ecosystem. Pathobionts, it is a very nice terminology. It means typically benign endogenous microbes with the capacity under the altered environment conditions to elicit pathogenesis. Prebiotics means nutritional substrates or fibers that promote the growth of microbes that confer health benefits in the host. Probiotics, live microbes that confer health benefits when administered in adequate amounts in the host. Symbiotic, its formulation consisting of combination of both prebiotic and probiotics. How can we analyze the gut microbiota? We have 6 techniques as shown in this table, and this table explains the advantages and disadvantages of each one of them. Microbial culture, but beware that the microbiota may be non-culturable. 16S ribosomal RNA gene sequencing, the most commonly used method to determine the abundance of taxa and diversity, because bacteria in the gut are diverse and rich. Limited taxonomic resolution analysis is a disadvantage, and you cannot differentiate between live and dead microbes. Metagenomic sequencing capable of determining the function capacity of microbial communities, but more sequences are needed per sample. Therefore, it is not cost effective. Metatranscriptomics, capable of determining which genes are active and turned on. However, as some proteins are regulated in a post-translational manner, RNA abundance can sometimes be uninformative. Proteomics, metabolomics potentially identify microbial molecules responsible for host pathological physiological status, cannot readily differentiate between host and microbial proteins and metabolites. Probiotics determine the functional impact of one or a community of microbes on specific host phenotypes. However, it is expensive. Germ-free mice have physiological differences compared to mice with a normal gut microbiota, which may influence outcomes. So, by fixing this slide, we can look at every method, every technique, advantages and disadvantages. Let us go to the correlation of gut microbiome with functions and disease associations. Here, gut microbiota is correlated with different functions because it influences immune maturation, host cell proliferation, vascularization, neurological signals, pathogen burden, intestinal endocrine functions, bone density, energy, biogenesis, and biosynthesis of vitamins, steroid hormones, neurotransmitters, and share metabolism, bacteria branched chain and aromatic amino acids, dietary components, bile salts, and drug and xenobiotics. So a lot of functions are affected and related to the presence of the gut microbiota. It's not only the different functions, but gut microbiota and its disturbance is associated with disease, neurological, psychiatric, respiratory, cardiovascular, gastrointestinal, hepatic, autoimmune, metabolic, oncology, and today I'm going to show some data about gut microbiome disturbance and kidney disorders. To start with, if we are here within the gut and we have normal bacteria, so the gut microbiome is in its normal situation, so the bacteria is symbiotic. Symbiotic bacteria, normal bacteria, lead to competition of four nutrients to the pathogenic ones, so it suppresses pathogenic ones, and as well it metabolizes dietary fibers to release short chain fatty acid, which are taken by the epithelia as food and share in the hemostasis of cholesterol and the glycogenesis. So everything is fine. The gut bacteria is within normal, epithelium normal, and metabolism is normal, and no inflammation. Everything is fine when gut microbiome is fine. There are some data correlating the disturbance in the microbiome and the risk of obesity and diabetes. Even bad effects, adverse effects of abusing fructose, because abusing fructose may lead to metabolic disturbance and type 2 diabetes. One of the pathogenetic mechanisms how abusing fructose leads to type 2 diabetes is through inducing dysbiotic gut microbiota by decreasing bacteroides, increasing firmicutes, and increasing gut permeability, with subsequent inflammation and endotoxemia that lead to insulin resistance, adiposity and obesity, metabolic syndrome, and type 2 diabetes. A very interesting study that was published last year about the correlation of early life antibiotic use that disturbs the microbiota, and the association of this disturbance of microbiota with development of obesity. The key point of this study is to answer a question: does antibiotic use in children during breastfeeding influence the long-term health benefits of breastfeeding? For the breastfeeding to be completed, it should be between 1 year and 1.5 years, according to this literature. But shorter duration of breastfeeding is not so protective, and even if we complete breastfeeding, however, the infant is treated with antibiotics, the use of abusing antibiotics in early life may lead to disturbance of gut microbiome and lead to obesity later on. So breastfeeding decreases the number of post-weaning antibiotic courses and decrease body mass index in later childhood. The protective effect of breastfeeding against the high body mass index in later childhood was evident only in children with no antibiotic use during breastfeeding period. And as I mentioned, breastfeeding should be between 1 and 1.5 years. The results suggest that the metabolic benefit of breastfeeding are largely conveyed by the intestinal microbiota, which is disturbed by antibiotic treatment, evident by lower abundance of this type of bacteria. Again and again, we should educate mothers to avoid abusing of antibiotics for their kids in early life. One of interesting issue is the gut microbiome interactions with drug metabolism, because gut microbiome can have all these biotransformation with drugs: reduction, hydrolysis, dehydroxylation, dealkylation, demethylation, decarboxylation, acetylation, deamination, deconjugation. And here we show you the different biotransformation, drug examples and metabolites, and the comments. You can fix and read everyone. I want here just to show you that diclofenac as non-steroidal anti-inflammatory drug and indomethacin is one of the biotransformation is glucuronidation, and some enzyme hydrolyze these compounds to release non-steroidal within the intestine, and this is the action of gut microbiome. Releasing of these drugs within the intestine lead to enteropathy. So if we think of this and we manipulate this metabolism through inhibition of this hydrolysis, we can protect the intestine against non-steroidal anti-inflammatory drug. A lot of biotransformation for different drugs and different comments, so you can go through them. Regarding the microbiota and the immunity, if we have normal gut microbiome, everything is fine, intestine is good, the immunological reaction is toward lower inflammation. However, if there is disturbance in the gut microbiome through different receptor, toll-like receptors, etc., using MY differentiation 88 or other pathways, it leads to shifting from Th2 to H1 and elaborating Th17, which is bad lymphocyte with inflammation, suppressing Treg cell. So if there is any disturbance in the gut microbiome, everything is moving toward inflammation. So if we have symbiotic gut microbiota, this means everything is fine. This will increase commensal bacteria, increase barrier integrity, and decrease inflammation. As you see here, through these interactions, the tight junctions are healthy, and here increasing of FOXP3, Treg cells, and lower inflammation. If we are dealing with dysbiotic gut microbiota, dysbiotic gut microbiota means disturbed gut microbiota, and disturbance means abnormality. So within the abnormal gut microbiome, you will find here the tight junction become non-tight, it becomes loose, with subsequent increasing permeability and shifting from non-inflammation to inflammation arena. You can find here activation of Th17, Th1 and other cells. So again and again, the presence of normal gut microbiome is one of the essential components for better immunological interactions. And within the transplantation environment, use of chemotherapy, antibiotics, immunosuppressants, post-surgical anatomy, disturbance, genetic factors, all these factors can lead to altered microbiome, with sequelae of this dysbiosis is adverse effects to the host and to the graft rejection, recurrence of underlying disease, host increasing infections, graft versus disease, immune complications, and increased mortality. Again, I'm just showing you that disturbance of gut microbiome can affect the immunity. A very interesting study showed that immune suppressive treatment altered the secretion of ileal antimicrobial peptides and the gut microbiota, and favor subsequent colonization by uropathogenic E. coli and infection. Here, just to show you that the prednisone will lead to more and significant difference in the microbiota, as you see, with increasing firmicutes and decreased bacteroidetes by using prednisone. So this is to consider the use of immune suppression, its effect and shifts in microbiota. Another study correlating the tacrolimus dosage with certain gut microbiome. The abundance of this type of microbiome leads within the intestine leads to the enhancing the tacrolimus and necessity of increasing the dose. So escalating the need of dose escalation. This means that the persons who are harboring this abundance of microbiome need larger dose of tacrolimus. What's meant by gut-kidney axis? It's a very nice axis, and it was explained very nice in this chapter. We can look at this just to know that we have a rich microbiome within the gut, and the color code here is reflecting the abnormality within the gut microbiome in a patient with chronic kidney disease. Red color means the bacteria or the gut microbiome is reduced. Green color means it is increased. So either decrease or increase are abnormalities. So you can look at the type of the gut microbiome and just look at the color code. Again, red means decrease, green means increase. Again, we have rich microbiome, different bacteria and different organisms within the gut. Here I just want to say that our gut harbors more than 100 trillion bacteria within the gut. So, and this exceeds tenfold the number of all human cells. So we are dealing with a very enormous amount. Just in a simple manner, in chronic kidney disease person and patient, all these gut microbiome are increased and all these are decreased. So I want just to fix a certain statement that chronic kidney disease is associated with abnormality of gut microbiome. This means dysbiosis. It's not a simple relationship, because disturbed gut microbiome will lead to losing of tight junction and increasing bacteremia and toxemia, uremic toxin release because of microbial origin, because the bacteria leads to metabolism of fibers and diet to release uremic toxins, and this will lead to inflammation. Inflammations will affect cardiovascular system and affect more and more the kidney. So dysbiosis can lead to chronic kidney disease. The environment of chronic kidney disease is associated with dysbiosis, abnormal gut microbiome. This means that the relationship between chronic kidney disease and the gut microbiome is bidirectional. Chronic kidney disease leads to gut dysbiosis, and gut dysbiosis leads to chronic kidney disease. A very interesting cohort study. 3.5 million persons were followed up. The presence of constipation is bad and associated with higher incidence of chronic kidney disease. Here, if we look at no constipation and constipation, the presence of constipation is associated with significantly higher percentage of chronic kidney disease and end-stage kidney disease. More importantly, the higher the degree of constipation, the higher the incidence, cumulative incidence of chronic kidney disease and the end-stage kidney disease. So this just to convince you by there is gut-kidney axis. So constipation is considered one of risk factors for chronic kidney disease. This is a very nice randomized control trial assessing this drug, which is lubiprostone, which is a colloid channel activator in the gut. It will lead to improvement of diarrhea, constipation. And this drug is used in this study just in adenine-induced chronic kidney disease model, experimental model, to alter the intestinal environment and to address if this altering of intestinal environment by using this chloride channel activator has any good impact on the kidney. If we look here, this is the control, normal control. This is the renal failure model, and with this is the renal failure model and use of the drug. What is your observation here in the Mason trichrome? This is fibrosis, disturbed glomeruli, and here significantly the use of this drug significantly lowered the chronic changes and chronic kidney damage. So this means that altering the gut microbiome by using this drug is beneficial in this scenario. Another issue, and this is very fascinating study because of using the marker studies, here the authors prove that autoimmune disease is exacerbated by Th17, which emerge and egress from intestine. How they proved by the experiment by giving color code and the marker, and here the photo labeling intestinal cells. The authors proved that microbiota induced Th17 cells egress from the gut and infiltrate the kidney via this receptors, chemokines, in immune-mediated diseases. This finding will build the basis for therapies targeting the intestinal Th17 cells reservoir to treat extra-intestinal Th17 autoimmunity. It's not only gut microbiome, but also disturbance of gut microbiome stimulate IgA production, not in a normal amount, but also in abnormal amount, with subsequent deposition within the kidney and to leading to IgA nephropathy. So again, uremia is associated with intestinal dysbiosis with subsequent releasing toxins that disturb all the health, CNS manifestations, protein energy wasting, immune dysregulations, cardiovascular disease, and the progression of chronic kidney disease. All these sequelae were related to dysbiosis. Again, the relationship between kidney and the gut is not simple, it is bidirectional. So both dysbiotic gut microbiome and chronic kidney disease lead to each other. This is a nice study. In this study, the authors proved that reduction in butyrate producing species of this gut microbiome is associated with chronic kidney disease progression. How they proved? You can find this very easy here in the correlation analysis. There is inverse relationship between inflammatory markers in the C-reactive protein and this type of bacteria. So the higher this gut microbiome species, the lower the C-reactive protein, because it is inverse relationship. And the higher this species, the lower cystatin C, BUN, and creatine. This means improvement of kidney function. And with the relationship is positive with estimated GFR. This means improvement of kidney function. So again there is another proof for the correlation of disturbance of gut microbiome and chronic kidney disease. Just to show you, the short-chain fatty acids elaborated from the gut microbiome, because gut microbiome metabolizes the fibers, releasing short-chain fatty acids. There is here you can find different actions of short-chain fatty acids, and I'll discuss the role of short fatty acid later. But here, just to show you that it is not a simple relationship between short-chain fatty acids and the actions, because here, when it bind short-chain fatty acids bind G related protein receptor 43, it will lead to decreasing blood pressure and here protection, improving metabolism, inhibiting accumulation of fat in the adipose tissue. However, if short-chain fatty acids bind G protein receptor 41, the short-chain fatty acids will stimulate the sympathetic nervous system and increase blood pressure. And if short-chain fatty acids bind olfactory receptor 78, it will lead to stimulation of renin release from afferent arteriole, increasing blood pressure. So it is not a simple, it is a complex actions. Another experimental animal, this is 5-6 nephrectomy rat model, which reflects chronic kidney disease. Here they prove that if we create this model, this will be associated with disturbance of gut microbiome by where they notice the reduction of lactobacilli and increasing bacteroids. This means that chronic kidney disease is associated with dysbiosis. And when the rats in these models are treated with retention enema, including lactobacilli, this treatment ameliorates the gut microbiome abnormality with subsequent improvement of kidney parameters. This means that gut microbiome is essential for the pathogenesis of chronic kidney disease. Even in hemodialysis, disturbance of gut microbiome is correlated with inflammation, and abnormality of gut microbiome may be related to dialysis-related factors and non-dialysis-related factors, and so the barrier is affected by all these processes. So the ischemia, reperfusion, hypoxia, a change in core temperature, urea-induced disturbance, gut edema, modification of the composition of microbiota, acute change to intestinal barrier, all these will lead to more disturbance of the gut barrier that is ameliorated by low ultrafiltration rates and cold dialysate, etc. Again, if we are thinking of gut microbiome disturbance, and we can find here that there is loosening of the tight junction with disturbance and development of inflammation within the chronic kidney disease environment. The relationship of gut microbiome disturbance and stone was discussed in many articles. It's one of the nice review that reviewed this issue, where Bacteroides genus was most abundant in the kidney stone group, and the E. coli inversely correlate with citrate levels. And we know that the depletion of gut microbiome is not an easy task. It can be depleted by antibiotic use, and here this just to show that the issue of gut microbiome is very complex. Here depletion of gut microbiome protects against renal ischemia reperfusion injury. So we should think of the issue as a complex, and to refine the research in this sector. Also, gut microbiome may be associated with chronic prostatitis and pain. So Up to this point, we discussed together the definitions and the correlation of microbiome disturbance with obesity, metabolic syndrome, immunological derangement, increasing inflammation, and association with many kidney disorders, including chronic kidney disease, stone, and ischemia reperfusion injury. I'm going to show in the coming sector how microbiota helps in generation of uremic toxin. This is a source from the diet: tryptophan, phenylalanine, choline, phenylalanine, and others. I just give an example of the sources for the uremic toxins. Induced sulfate, the source from tryptophan, and it's protein-bound toxin, and tryptophan is metabolized by gut microbiome, and then liver elaborate induced sulfate. The same tryptophan may lead to indole 3-acetic acid by other bacteria, phenylalanine and tyrosine to bicrystal sulfate, choline to this one, phenylalanine to this one. So this is a source, and this is the name of the toxin. Here, as I mentioned, the gut bacteria or microbiome metabolizes the original source to release the indoles, bicrystal, TMA. This is further refined by liver to be released as toxin, induced sulfate, for example, bicrystal sulfate, into the circulation. Okay. Again, here, if we compare non-dialysis to hemodialysis patients, you can see here the difference. Significantly higher levels of uremic toxin in the hemodialysis patients. Induced sulfate, bicrystal sulfate, and indolacetic acid. This means that there is disturbance of gut microbiome. The release of uremic toxins are bad because, and this is one of the problems created by disturbed gut microbiome within chronic kidney disease and dialysis patients. Induced sulfate has very enormous adverse events: inhibit endothelial proliferation and repair, increase endothelial leukocyte adhesion with subsequent atherosclerosis, promote cardiac fibrosis with subsequent congestive heart failure, increase arrhythmogenesis with development of arrhythmias, increase vascular smooth muscle proliferation, increase vascular calcification, arteriosclerosis, impair new vascularization mediated, and this will lead to peripheral arterial disease, increase expression of tissue factor with subsequent vascular access thrombosis. So here, this slide summarizes all bad effects of induced sulfate on the cardiovascular system. Again, impair the gut barrier because of the presence of inflammation and gut dysbiosis that lead to fermentation and releasing of toxin, can increase CKD progression, cardiovascular disease, protein energy wasting, immune dysregulation, and CNS toxicity. I think I repeated this statement by many ways just to consolidate the gut-kidney axis and the problem of gut microbiome. How to manipulate gut microbiome? This is one example of uremic toxins that we may think how to manipulate by altering degeneration through increasing carbohydrate fiber, but be aware of risk of hyperphosphatemia and hyperkalemia in CKD patients, to decrease protein intake, but be aware of the risk of malnutrition, using pro-living, pre-fiber, or both pre and probiotics by increasing saccharolytic activity of bacteria, using laxative to decrease colonic transit time, using adsorbative oral adsorbent like ACT 120. However, when it was tested in larger study, it was negative. It doesn't lead to beneficial effects as we think. So again, this means that we should think of multiple modalities preserving kidney function to improve the transporter expressions, and the refine dialysis by combining convective and diffusive removal, and ultimately renal transplantation. So this is our thinking. If we start the journey of manipulation of gut microbiome by prebiotics or diet, one of the important source of prebiotic is the encouraging healthy foods, increasing fruits and vegetables and fibers, because dietary fibers have many beneficial effects. Here they reduce dietary acid load. To the extent if we start fibers and vegetables earlier in chronic kidney disease, it is equivalent to supplementation of sodium bicarbonate. Stimulates saccharolytic fermentation, releasing short-chain fatty acids. Short-chain fatty acids have good functions and reduce inflammation. And here inhibit suppress proteolytic fermentation, has antioxidant effect, and all these beneficial effects lead to decreased cardiovascular disease. This is to confirm the Hippocrates statement, let food be the medicine and let medicine be the food. This means that healthy food is very important for the healthy life. Again, according to the issue of the American Journal of Kidney Disease of March 2017, this month, dietary modeling of foods for advanced chronic kidney disease based on general healthy eating guidelines. What should be on the plate? What should we eat? It's a very nice and very interesting question. We should encourage healthy foods, healthy diet, increasing plant-based foods in the early part of the chronic kidney disease, because in late part we are afraid of gastrointestinal upsets and hyperkalemia, and to reduce salt all through, because reduction of sodium is beneficial for chronic kidney disease and helps in preserving kidney function. So following this good habit of diet, it will help in reducing blood pressure, reducing proteinuria, reducing the release of uremic toxin, reducing acid load, reducing rate of falling GFR, and reducing inflammation. Again and again, we should encourage healthy food to our patients. This is one of the experiments showing the role of high fiber diet and assist supplementation. It changes the gut microbiota and prevent the development of hypertension and heart failure in mineralocorticoid salt hypertensive mice. If you look here, in comparison to the positive control, the use of fiber ameliorate histopathological damage in the kidney. This means high fiber diet is essential. Again, the gut function, kidney function, liver function are improved by the high fiber diet. The use of prebiotic in general, we need to focus on evidence, because using this prebiotic arabinoxylan oligosaccharides on microbiota derived uremic retention solutes. This was a randomized control trial including 40 chronic kidney disease patients with estimated GFR between 15 to 45 milli per minute. This study has negative evidence, so it shows no difference by the use of this prebiotic. So again, although the thinking of this issue is fantastic, however, we need a lot of evidence to convince us by using certain prebiotic. But to encourage fibers is very fine and very nice. Regarding the role of short-chain fatty acids, short-chain fatty acids come from the metabolism of fiber in the diet, and we have many short-chain fatty acetate, propionate, butyrate, and all these are the short-chain fatty acid. Here, through the monocarboxylate transporters and other transporter, short-chain fatty acids helps in keeping the tight junction. If you compare here, the disturbed gut microbiome lead to disturbance of tight junction. The using of short-chain fatty acids improve tight junction. So all the beneficial actions, inhibition of endotoxin, regulation of immune response, anti-inflammation, amelioration of kidney injury, can be the result of the beneficial effect of short-chain fatty acids. Short-chain fatty acids bind G protein, different receptors, and here we have B-arrestin. So thinking of this molecule, refine the research, how to use this molecule in the medicine and in the research, and to establish drugs targeting all these molecules maybe in near future in the treatment of the chronic kidney disease. So we are needing a lot of research. Short chain fatty acids were tested in many problems related to kidney disorders like acute kidney injury induced by ischemia reperfusion injury. In here you can look at the route of administration and dosage, contrast-induced nephropathy, the gentamicin-induced nephropathy. All these three diseases sectors were ameliorated by the use of short-chain fatty acids, and you can read in detail. However, urethritis and hydronephrosis are worsened by short-chain fatty acid. This and improved by using mTOR inhibitors. This means that it is not a simple relationship, and sometimes you may find conflicting data. So what we need is more refinement, more understanding how to manipulate the axes. So dietary fibers leading with the presence of gut microbiome release short chain fatty acid with subsequent binding to different receptors leading to different actions. So we need to refine to use it properly. So this is regarding prebiotics. What about probiotics, which are a living organism taken this slide summarizes different commonly probiotic formulations, either using single organism or composite probiotics. You can look here and to the different generation, and this is the different actions, or as I mentioned in the prebiotics, beneficial effects on everything. Here the same. Probiotics maintain barrier function, enhance the microbial flora, have a good effect on the innate and adaptive immunity, modulate signal transduction, nice metabolic effects, colonization resistance. So a lot of beneficial effects can be produced by the use of probiotics. Oxalobacter formigenis, all of us are aware by the value of Oxalobacter formigenis in combating oxalate stones, and this very nice experimental study targeted the mechanistic approach, how Oxalobacter formigenis deal with the oxalate. They proved that Oxalobacter formigenis stimulate oxalate transport by intestinal epithelial cells. This means when it's increased secretion of oxalate by the intestinal epithelial cells, it will reduce oxalate load to the urinary tract. This means reduction of stones. However, the oxalate stone is not restricted to Oxalobacter formigenis, but disturbance of Lactobacilli and other bacteria also lead to oxalate stone. However, all this table summarizes some studies which were carried out to prove the efficacy of Lactobacillus containing probiotics for treating hyperoxaluria, and unfortunately the majority of these studies were disappointing. Regarding the effect of dietary yogurt, and the dietary yogurts can affect the healthy human gastrointestinal tracts, and we can encourage the use of healthy the yogurt in early phases of chronic kidney disease. And the yogurt may be fortified by lactobacilli or probiotics, and the consumption of probiotics and yogurt has beneficial effect on kidney function. Again and again, we should be aware, because the use of these supplements of probiotics and herbal remedies, all of these are considered complementary medicine and not subjected to the full regulation and administration done for drugs like FDA. Approval of any drug is very complex, but here you can find different formulations of probiotics that are not subjected to this administrative policies. So you should be aware of that. And this is an example of publication showing that commercial kidney stone probiotic supplements may be rubbish, because analysis of these commercial ones, the probiotic supplements sold over the internet by ProLab Limited and Sanzyme Limited didn't contain identifiable Oxalobacter formigenis or viable oxalate degrading organisms. This means that it will not be of any benefit. Synergy, it is combining prebiotic and probiotic. That's one of the nice study, including 37 patients with chronic kidney disease pre-dialysis with estimated GFR between 10 to 30 milli per minute. And the study was done in 2 periods with 4-week washout period. And the study proved that the use of synergy, prebiotics and probiotics, especially in a patient with who were free from antibiotic use, so avoiding antibiotic use, was this habit using prebiotic, probiotic, and avoiding antibiotic. And this study showed significant reduction of uremic toxins, either total induced sulfate, bicrystal sulfate total, or free induced sulfate and bicrystal sulfate. If you compare treatment versus placebo, you can find significant difference, significant p-value. So again, we should think of issues. Even in dialysis patient, using probiotic supplementation for 60 diabetic patients in a randomized control trial was proven to be beneficial with the reduction of inflammation in dialysis patients. Again, so I discussed how to manipulate by using prebiotic, probiotic, symbiotic, and here I'm going to say that probiotic may be administered through genetic engineering. So this is one of the studies showing that amelioration of cadmium and mercury induced liver and kidney damage in rat by genetically engineered probiotic E. coli Nicel 1917, producing quinone with oral supplementation of citric acid. And a nice, very nice report, brief report, how to modulate a circulating uremic solute via rational genetic manipulation of the gut microbiota. Here, this is the study in very brief, in just a figure. In a brief, authors identify a widely distributed family of indole-producing tryptophanase and commensal gut bacteria. Using this information, they engineer bacteria to control in vivo production of the downstream product, the uremic toxin induced sulfate. This result suggests a new strategy for treating chronic kidney disease by targeting the microbiome. So, and these are the highlights of this study. Indole-producing tryptophanase in bacteroids are identified computationally. The kinase activity and its control of induced sulfate levels in vivo are established. Rational diet alteration in gerontobiotic mice with a synthetic community control induced sulfate. Colonization with kinase deficient bacteroids lower induced sulfate in conventional mice. So I discussed prebiotic, probiotic, symbiotic, genetically engineered probiotics. The last resort may be to transplant fecal microbiota. So fecal microbiota transplantation is emerging and is a relatively new modality, although it's not widely accepted. However, it is increasingly used in Clostridium difficile infection refractory to the standard of treatment. And here, if you look, nowadays they are calling for the national registry to be an important step toward understanding risks, benefits of microbial therapeutics, because donors should be selected properly, subjected to thorough investigation, including viral, because to avoid infection. And the gut microbiota is transplanted through different routes, colonoscope, enema, and nasodidenal, but it's not commonly used nowadays. Just to convince you by this modality, this is one of the studies discussing the difference between donor or autologous, allo or auto use of fecal microbiota in a randomized control trial to treat Clostridium difficile infection. And here, donor stool administration via colonoscopy seemed safe and was more effective than autologous fecal microbiota transplantation, preventing further Clostridium difficile infection episode. And this is infection versus frozen versus fresh microbiota. Here the study included, for me, it's considered large number of patients, 114 and 118. All of them were treated by fecal microbiota transplantation, either frozen or fresh. And this study showed that frozen may be a good alternative to fresh. This is the last study I found on the internet that was published in February this month, last month, on the Lancet, multi-donor intensive fecal microbiota transplantation for active ulcerative colitis, a randomized placebo-controlled trial including 85 patients. And it seems that the use of fecal microbiota transplantation is beneficial in this disease. So up to this point, I finished definitions, the microbiome and its relation to different diseases, and I explained to you gut-kidney axis, and then how microbiota released uremic toxin, and then how to manipulate gut microbiome by prebiotic, probiotic, symbiotic, genetically engineered probiotic, and the last resort may be fecal microbiota transplantation. I want to finish this presentation by just stating the fixed rule with dealing with gut microbiome to treat diseases still in its infancy, and we need a lot of research, a lot of refinement, a lot of convincing, a lot of data. But currently we are believing that disturbed gut microbiome may be associated and linked to many disease disturbance. The correlation of gut microbiome with chronic kidney disease, yes, there are many challenges to overcome, and it may, when we think and we refine this relatively new arena, we may improve the disease progression. Nobody knows. It may be in the future. The treatment of chronic kidney disease may be through manipulating gut microbiome, at least to decrease uremic toxin or to decrease the progression of chronic kidney disease. And how to purify these issues and to deliver it in a very pleasant way, this will need a lot of research. At this point, I think I finish, and so thank you very much for your attention, and I'll be very happy to receive your any questions in future. Please go to our website, SNT Virtual Academy, and the module of the MD nephrology, and answer the mock exam on gut microbiome and kidney disease. Thank you very much. Bye bye.